Hi, everyone. Just in case you're watching this in the future, this is Mr.Dealva from the past. Oh, look, there's like a little. Whoa! Anyway, why does my hair really look like that? Incredible. It's like falling out. All right, we're coming up. Yeah. Okay. For those of you guys who don't have long hair, it's pretty cool to be able to do that. Anyway, sorry. That doesn't need to be part of the recording. Okay, histology is the study of tissues, and tissues are collections of similar cells and the substances surrounding them. We'll talk about extracellular matrix when we talk connective tissue. But remember our levels of organization, okay? Starting at atomic, molecular, cellular, tissue, organ system, organism. Atomic and molecular, that's medical chemistry. So we're not really going to get into atomic or molecular unless it's very mandatory, because you should have gone over that. Cellular is pretty much all AP Bio. AP Bio is pretty much all cellular biology. If you didn't take AP Bio, that's okay. You'll be okay for our purposes. Anything that's super important at the cellular level, I will tell you about. Like, there's a little bit we'll go into about the cellular level today. Obviously, histology, being the study of tissues, we're focused primarily on tissues. Because we need to know what tissues are before we start talking about organ systems. Every unit after this in our class will be organ systems. And in those organ systems, we'll talk about the individual organs. And to understand that, we kind of need to know the tissues. And then we'll talk about all the organs in that system, how they work together. That's the organ system. And then by the end of the year, hopefully we've covered all of the organ systems, which then makes the organism. And then we've covered everything, and we're all good. And you know everything about the human body. Questions, comments, concerns? Okay. I will tell you what's mostly important to know for these slides, but stay with me. You do need to know. I need to move my little face. There are 4 types of tissues. Everything in your body primarily is made of 4 types of tissues. Epithelial, there's connective, nervous, and muscle. How do I get this to go away so that you can see stuff? Let's do it like this. Whoa, it's freaking out a little bit. There we go. That should do it. There we go. Okay. Today, and probably tomorrow, we're just going to cover epithelial and connective, because there's plenty to know within just those 2. We'll cover nervous tissue when we cover the nervous system. We'll cover muscle tissue when we cover the muscular system. Okay, questions, comments, concerns on this? No. Again, if I'm going too fast, please slow me down. Maybe you should do like a little hand signal for slow the heck down, Mr. Dealva. Yeah, there you go. Stop, or time out, or stop. Either of those will work. All right. So today we're literally just going to spend the entire period talking about epithelial tissue. Okay. Tomorrow we're going to talk connective tissue. All right. So just kind of an overview. Epithelial tissue covers stuff. It is the surfaces and the linings of your body. It is the surfaces and the linings of your body. So every outward surface of your body, that's epithelial tissue. So your integumentary system, you two in the back? Yes. I guess, Coral, you didn't do integumentary. It was in Selena. That's right. Integumentary is pretty much all epithelial tissue. Integumentary is basically all epithelial tissue. But we'll talk more when we talk, that unit. Okay, this diagram is really funny, but it's also important to know that all of the surfaces, so all of your outside skin, all of your serosa, right, parietal, visceral serosa, but like even anything that could be considered the outside of your body, like even the inside of your mouth, all the way down that pipe into your stomach and then the intestines, that whole tube connecting you from mouth to butt, that's all epithelium. The epithelial tissue is the barrier between your body and the outside world. So if you ingest something and it's in your stomach, it still has to go through epithelial tissue to get into your body, right? And that's either being absorbed in the large or small intestine, right? That's going through the epithelial boundary, okay? So that's kind of the defining characteristic of epithelial tissue. It is the boundary between your body and the outside world, right? But you can define the outside world as like even the inside of your stomach. Right? If something's going to get into your body, it has to go through epithelial. Epithelium. I'm going to refer to epithelial tissue as epithelium. Those two can be used interchangeably. Epithelium is the same thing as epithelial tissue, just so you know. You're going to have to excuse my incessant kind of need to continue talking, even when we're just waiting. Okay. Again, slow me down if you need to. All right. There's also glandular epithelium, which we're not going to get into too much depth, but you have endocrine and exocrine glands. These are your exocrine glands, but we're not going to get super into it. You just need to know that it exists. Glandular epithelium. We've probably heard of the endocrine glands because those are... Oh, I did this again? I have to... Oh, no, it works. Cool. We'll talk more in the endocrine system, but your sweat glands are also technically part of your epithelium. If you think about that surface, it like goes inwards, and that's the gland, so it's epithelial tissue by necessity. Okay, you don't need to know the specifics of this. I'm not going to test you on glandular epithelium, but you should know that it exists. Okay, moving forward, because we have some stuff to cover. Okay, here's something else you need to know. Epithelial tissue is on the outermost surface of your body, whether that's like literally surface layer on your skin or inside of your mouth or inside of your esophagus or inside of your stomach. That is considered facing outwards. So 2 words that you need to know. You need to know apical versus basal. The apical surface is the one facing the exterior, whether the exterior is the inside of your stomach or whether the exterior is just the outside world, outside of your skin. And then you have the basal surface. Think of that as like the base. The basal surface is where your epithelial tissue is connected to your connective tissue, and then therefore the rest of your body. You need to know the difference between these 2 words. At any point, if you have questions, please raise your hand. Okay, you need to know apical versus basal. Okay, I'm going to put this next slide up here for a second, and then move past it and come back to it just so you have a little context. Okay, you need to know microvilli and cilia. Microvilli and cilia, let me show you what I mean. Eyes up front really quick. Okay, so the apical surface can be smooth, kind of like our skin. I mean, our skin has hair follicles, but it can be smooth, or it can be covered in microvilli, like in your intestines. Matthew knows this, which is for secretion absorption, or it can be covered in cilia, like in our trachea or in our bronchioles, and cilia kind of move all together. But just so you know what those are, you need to know what microvilli are and what cilia are. You don't need to know these specific definitions, but you do need to know what they are. Sorry. Oh my gosh. I cannot do this. There we go. Also, you have access to these slides. They are posted in Schoology. My prediction totally getting worse. Very nice.
Okay.
I wonder if I can pause and play this recording so that when you're watching it back, you don't have to wait for me to wait for you. So like this is the apical surface of your epithelium out here. And the basal surface would be the opposite side of those cells that is connected to the basement membrane. Yeah, we'll talk about that. Yes, good question. Okay, I'm going to move forward. Yes, we'll talk about that more in the integumentary system. Okay, so as previously said, the apical surface can be smooth. It can be covered in microvilli. I want you to think about how that changes the surface area. Just like our digestive system people said in their presentation, it increases the surface area by a lot. If you think about the amount of paper it would take to cover like my arm, that's not a whole lot if you think about paper as surface area. But if you had to go over a bunch of ridges and valleys, it increases that surface area by a large margin, right? So instead of one piece of paper, it might take me 5 or 6 pieces of paper to cover all of those projections. I thought I did a little drawing of it, but that's okay. Questions on microvilli? Very good for secretion or absorption, right? If you think about fitting little pores on a piece of tissue that can absorb nutrients, it makes sense to have as much surface area as possible. So having more surface area allows for more pores for absorption or for secretion. Cool? Okay. Cilia are kind of like these little hair-like projections, and they kind of beat all together. If you've seen like a little microorganism swim around, a lot of them have cilia, and they all kind of do that. That's all happens in your trachea and in your lungs. The cilia kind of sweep together all the dust and particles and push them into one little pile, and then you can cough up that mucus. So just the same way that you sweep things into a pile before you can collect it, your cilia kind of push things around. Does that make sense? Okay, moving on here. We need to know the basal lamina. Now, this is a lot of information. Let me give it to you in context briefly. The whole idea of the basal lamina is that you have your epithelial cells, and they are connected to this basal lamina, and that's part of the epithelial tissue. It's like the base structure, basal lamina. That basal lamina is connected to connective tissue called the reticular lamina, and together that makes up the basement membrane. So what you really need to know is that the epithelial tissue is connected to the basal lamina, which connects to the reticular lamina, and that's called the basement membrane. Epithelial tissue connects to the basement membrane. I'll put that back up here. I will give you a few examples. Oh, yeah, I guess that's fair. Think of it as just the way that your skin is attached to you. There you go. Yeah, it's like you're gluing the cells down. It's like they're holding on. They have little anchors. That's the basal lamina. And the basal lamina is like grabbing onto the connective tissue that is the reticular lamina. And together that makes the basement membrane. So we just refer to it as the basement membrane. It's like how they attach to your body. That's why your cells don't just fall off, which is not something we typically think of. Okay, questions on this? You don't need to know this in extreme detail. You just really need to know that the basement membrane is what attaches your epithelial tissue to your body. You know what? I am going to pause the music because I'm worried that it's going to mess up the recording. Sorry. Okay. All right, I'm going to push us forward a little bit. What you need to know here is basically on this slide. You need to know that the cells that make up your epithelial tissue attach to the basal lamina, which attach to the reticular lamina. And together, those are the basement membrane. Basement membrane is the thing that holds your cells onto your body. Okay, move in forward a little bit more here. We're going to talk about how these cells are held together laterally. Okay, let's look at these cells one more time before we talk about this. We have our, what's this surface called? Apical surface. And you have the basal surface. Okay, each of the sides of the cell are called the lateral surface. You don't need to know that, but how are these cells held together? How come they don't like come apart? Why can't I just like spread my skin apart? Right? I know it's kind of weird. Sorry. They are connected with 3 different things. They are connected with, well, there's actually more than that, but desmosomes, tight junctions, gap junctions. And they look like this, but you don't need to know it in detail. The extent of the detail you need to know is entirely on this slide. So you need to know desmosomes, they kind of like anchor tight junctions, are like tying them off, blocks the flow of fluid, and gap junction allows transfer of fluid between cells. And we'll talk about those in a little bit more detail, but this is the extent of what I need you to know about those 3 things. Did you cover this in AP Bio? No? Okay. In just regular bio. You didn't learn about birds in AP Bio. Yes. Regular bio was freshman year. AP Bio would have been last year for you, probably. Desmosomes. Okay. I am taking a video. Hi. Okay, I'm going to move forward a little bit. Okay, desmosomes. So if you're looking at this image and a little confused by it, this surface right here is the plasma membrane of one cell. So it's like this surface right here on the side, and then here's the other cell. So this darker material in between is between the cells. It is the intercellular space. Desmosomes, they really look like that. They're kind of weird. They basically just anchor onto each other. It's like they're holding hands. It's how the cells stay together. They're anchored together. Now, stuff can still flow between the intercellular space, but it's basically just holding onto the cell next to it, and that allows your skin to not come apart. Cool? Then you have things called tight junctions, which also hold your skin together, or hold your cells together. Tight junctions are called tight junctions because they basically pull the cells together so tightly that fluid cannot pass. That's why they're called tight junctions. Fluid cannot pass between cells like this. They cannot pass through the cells. Also laterally, stuff from one cell can't get into another cell through a tight junction because it's just like a protein. Think of it like a rope, and it just like ties them together very tightly. Questions on tight junctions? Okay, then we have gap junctions. Gap junctions are like little pores, little channels that go from one cell to another. So stuff can actually flow between your cells, which is pretty cool. If you've ever lived or been anywhere where they have actual weather, like when it snows, you'll see that sometimes downtown they have like buildings across the street from each other, but there's like a little walkway attached that you go in the building and you walk through this little walkway that connects the buildings so that you don't have to walk outside in the weather. That's the same kind of idea as a gap junction. It allows transfer of ions or nutrients between cells. Questions on gap junctions? Okay, gap junctions also allow exchange in the extracellular fluid. So it's not blocking the passage of extracellular fluid. Just the tight junctions do that. Okay, questions on this? Cool. That's the extent of what you need to know for the junctions. Okay, let's talk. 2 more terms you need to know. You need to know avascular and innervated. Epithelial tissues are avascular. That means they are not serviced by blood vessels. Okay, vascular refers to your blood vessels. Specifically here, we would be talking about your capillaries. Avascular means without blood vessels. So blood vessels do not bring oxygen and nutrients to these cells. We can also say that these epithelial tissues are innervated. They are innervated, which means that they have nerves. That one's easy to remember because it has nerve in it, innervated. There are nerves in your epithelial tissue, which is important because you use your hands to feel things, right? You need those sensory nerves, sensory neurons in your outermost layer of your body, in your epithelial tissue. So you need to know what avascular means. You need to know what innervated means. If I told you that a tissue or an organ was vascular, what does that mean? It is supplied with blood, yes. Perfect. Here, epithelial is avascular. That's one of the characteristics of epithelial tissue. They're avascular, but they're still innervated. You might be wondering, how then do they survive if they are not supplied with nutrients and blood? The nutrients and the oxygen diffuse through the connective tissue and the extracellular matrix or extracellular fluid. So blood vessels come, let's say the blood vessels come up to like here. The oxygen molecules and the glucose molecules can diffuse up towards these cells. So they still get the oxygen and they still get nutrients, but not directly from the blood. Does that make sense? Cool. You need to know that they're avascular, right? But that also implies that you don't have blood vessels that reach every single cell of your body. That would be too complex. So some things can diffuse. Good question. Okay, moving on. Let's talk about, I think characterizing is next. One last characteristic that I want you to know is that epithelial cells regenerate very quickly. They regenerate quickly, which is important because they are frequently exposed to friction and injury, right? They are the barrier between the outside world and your body. So they're pretty expendable, right? Because the outside world tends to damage your inside world. So that's another characteristic, right? Your nerve cells do not regenerate very quickly. Epithelial cells do. And it kind of makes sense why they need to. Questions on that?
No.
Cool. All right. Last thing I'll say on overall epithelial tissue. Just as always, anatomy, physiology. Structure dictates function. Okay? The structure of each specific tissue determines the function. Right? There are a lot of different functions of the epithelial tissue, and depending on the type of epithelial tissue, it'll have a different structure, which informs its function. For instance, your skin is not covered in microvilli because you don't need a lot of surface area. It's for protection, right? So the structure informs the function. A lot of different functions of the epithelial tissue, like protection, absorption, like in your guts, filtration and excretion. Filtration, like your kidneys, excretion, like your glands, secretion, also like your glands, but your exocrine glands, and then sensory reception. So that's why it's innervated, right? You can feel things. Questions on structure or function? Okay, let's talk classification. I'm going to pause on this next slide for a while and draw some stuff on the board to give you a little context. But here we have the different classifications of epithelial tissue. So there are two main ways that we categorize types of epithelial tissue. One is by the number of layers of cells. So that's simple stratified or pseudostratified, which is up here. We also have the shape of each of those cells. So you have squamous, which is a wonderful word. Squamous, you can also pronounce it squamous. I've heard both. Squamous, flat cells like this, cuboidal, cube-shaped, or columnar, which is wonderful that they're actually named after what they look like, which is unfortunately rare in science. Scientists like to name things after themselves. which is unfortunate. It makes more sense to call something cuboidal if it's a cube shape. I suggest you draw these. Columnar and squamous. Squamous. Okay, I don't know how well we can see these things on the... I know, I want to make sure that they can see. They can't see very well. There's pictures in the slides, you'll be all right. Squamous, cuboidal, columnar. Simple, stratified, pseudostratified. Let's talk about this in a little bit more depth. Is everyone good up here? No, okay, no worries.
I think she like the notes at home.
What's up?
Nothing.
All right, you are more than welcome to take pictures of this. If you want to do that after, I'm not going to erase anything, or I might add more stuff. So if you want to do that after class, you're welcome to take a picture of this stuff. Or you can just draw it in your notebook, either way. Okay, so very straightforward over here. Squamous cells. Here, let's start with a number of layers in cells. Okay, we have simple and stratified. These are very easy to understand. Simple is one layer of cells, and each cell in that tissue is attached to the basement membrane. The basement membrane I drew here in orange. I'm going to move this a little bit, just for right now. So, simple, one layer of cells attached to the basement membrane. Stratified is several layers of cells, and the defining characteristic of stratified is that some of these cells are not attached to the basement membrane. They're not attached here. They're attached to the other cells. And that's the defining characteristic of stratified. Okay? Pseudo-stratified, what does the prefix pseudo mean? Anyone know? It means fake. Pseudo means fake. So pseudoscience, fake science. Right? You are not going to cure any diseases by ingesting essential oils. Okay? I have essential oils over here because they smell nice. That's it. I like the way they smell. Okay? Pseudo also, there's a Pokemon named Sudowoodo, and he's cute. He's a little tree guy, but he's not actually a tree, he's a rock. So he's called Sudowoodo, and I find that funny. Pseudo-stratified. It's not actually stratified. It looks stratified, but it's fake, right? Every single cell here, and I actually drew the nuclei on these, every single cell is attached to the basement membrane. Even though they're kind of stacked on top of each other, they're not actually in layers because each one is attached to the bottom. Questions on pseudostratified. That one's a little tricky. Here's a picture of it. Here's pseudostratified epithelium. Right? Every single cell is attached to that basement membrane, right? Which is the basal lamina and the reticular lamina. Together make up that basement membrane, which I drew in orange here. We'll call this the basement membrane. Okay, how are we doing on time here? Crushing it. Cool. Questions on this? Okay, here's another look at squamous, cuboidal, and columnar. I always remember these because cuboidal is cube-like. If you're looking at an actual specimen, oftentimes they look round or spherical. Those are still cuboidal. Cells are just squishy, so oftentimes they don't hold up their form, especially if you have a sample that has been like stained and like put on a slide. They get a little bruised up, so they're not always perfect squares. In fact, cells oftentimes are hexagonal. You don't need to know that. Cuboidal are roughly even. Columnar, they're like little columns. They're pretty easy to identify. Perhaps the most easy to identify are the squamous. Squamous, I always remember because it's a silly word. I like to say squamous. And the squamous cells are kind of squat. That's how I remember it. Squamous cells are squat. They're flat, scale-like. Questions on these 3 different types of cells? Okay, these are epithelial cells. All right, not all cells fall into these categories. Okay, next, let's talk about classification. All right, so here we would say this is a simple what? Simple squamous, exactly. This is a simple squamous epithelia. Here, I would say this is a stratified cuboidal. It's kind of transitional. If you look at these top ones look maybe a little more squamous than the bottom. But here's just another image to look at. Squamous in 3D. Cuboidal in 3 dimensions. See how they're hexagonal? And then columnar, and here's what columnar look like. Whenever we're classifying epithelia, we take the first word to describe the number of layers and the second word to describe the shape. We put those together. So for instance, up here we have a pseudostratified columnar epithelium. I would also describe this as a pseudostratified columnar epithelium. Even though not all of them are column-like, they're not all columnar. This one right here might be going up backwards, right? You got to think about whenever you're looking at an actual microscope slide, you got to keep in mind that's a 2D image of a 3D object. So what plane are we getting? What slice are we getting? I'll talk about that when I show you actual pictures. Okay. So for instance, this I might call a simple cuboidal epithelium, even though they're circles. It's easier to draw circles. Here, what would this be? Yeah, first word, stratified, because there's many layers. Second, cuboidal, because they're cube-shaped, right? So this is a stratified cuboidal epithelium. You can say stratified cuboidal tissue as well. Okay, so you just kind of take the first one, put it together with the second. I will keep these like this so you can see. Let me swap these. No, it's fine. So simple squamous. You can have a simple cuboidal. You can have a stratified columnar. Well, you don't actually typically see those, but that's fine. Okay, let's take a look at some examples. For instance, here is a simple squamous epithelium. It's not the most ideal, but you can see this image is the alveoli. So who did that? That was Callie, and who else was?
Respiratory.
Yeah, who else did respiratory? Yes. Okay, so your alveoli are tiny little air sacs, and you can see that here. Each of these is an air sac, and they want those membranes to be really, really thin so that you can have more of them, and so that they can all come into contact with the air, and therefore oxygen, gas exchange. So here we have a simple squamous epithelium. It's simple because it's only one layer thick. It's squamous because all those cells are really thin, right? It's hard to even see the nucleus in some of these. And it's epithelial tissue. So simple squamous epithelial. Questions on this? Okay, let's take a look at some more examples. Here is a simple cuboidal epithelium. Now, very easy to see that it's simple cuboidal right here. A little harder to tell here. Remember, whenever we're looking at a 2D image, like a microscope slide, like for instance here, this just looks like a circle. We're looking at a cross-section of an alveoli, which is like an air sac. So when you get the cross-section of a sphere, it looks like a circle, correct? Here, we're looking at inside the kidneys. So let's say I have a tube, right? Because stuff goes through the tube in the kidneys. And if I cut it like this, like barely on the edge, it's going to look like a flat little piece of metal. If I cut it straight in the middle, like a coronal cross-section here, it's going to look like a rectangle, right? If I cut it like this, it's going to look like a circle. This is what we're seeing here. So we have the tubes in the kidneys cut this way. That's what we see here. And that tissue is one cell thick. Think if you take this and you kind of roll it up into a little donut, that's what we see here. Cool? If we're looking down here at this one, that looks like maybe a tube that was cut this way, and you can kind of see the inside of it. Does that make sense? Okay, it's always really important to kind of get your orientation, because we can look at stuff like this and say, oh yeah, that's a simple cuboidal epithelium. But that's not helpful if we don't know what we're looking at and in what orientation. Cool? Are we comfortable with why this is a simple cuboidal epithelium? Okay. Right here, this cell is cuboidal. This cell is cuboidal because they're not super tall, but they're also not super flat. They're cuboidal. And all together, that's one layer of, that's one layer of cells. Here is another sheet of simple cuboidal epithelium. So each of these tubes is like if you took this, we're comfortable with why this is a simple cuboidal epithelium, right? If you took that and rolled it into a tube, it's not one big mass of layers. It's one layer in a circle, right? And it's simple because it's only one layer of cells. Does that make sense? Helpful? Okay. Here we have simple columnar epithelium, which is pretty easy to identify. Each of these cells are kind of column-like, and it's one layer of them. This is in your GI tract. And you can see there's a little bit of fun stuff going on top of these. Those are the microvilli. So if you zoomed way in, you would see villi. And then on each of those villi, you would see microvilli to really drastically increase the surface area. Questions on simple columnar epithelium? Okay, these names can get pretty complex, but it's just piecing together all the things that define the tissues. So for instance, here is pseudostratified ciliated columnar epithelium, right? It's just adding more adjectives to be more specific. Pseudostratified is typically pretty hard to identify in an actual sample, but you can see that each of these cells actually does touch the basement membrane, which is down here. But it's tricky. Again, it looks kind of like this. And then the very top is covered in, kind of looks like a sea anemone, right? And the sea anemone have all those like little fingers. Those are the cilia. So you have the ciliated from up here, pseudostratified, columnar, because they're tall cells, epithelium. Questions on this? Okay, I probably won't have you identify different types of epithelial tissue with actual samples on a test. I would probably give you something like this, right? Because it's a lot more clear. Does that make sense? Okay. It is important to be able to identify these things, but we don't have enough time to practice it to a level where I would feel comfortable assessing you on that. Does that make sense? Okay. So you can put together basically any of these. So you can have a simple columnar, kind of like up here. Well, that's pseudostratified. You can have a stratified cuboidal. You can have stratified squamous. Our skin, any guesses what our skin is? It's stratified squamous. Right? Lots of layers of thin sheets. So the rest of these slides are examples. I'll let you go through them if you'd like. Stratified, we kind of already defined that. Here's stratified squamous, which is kind of like our cells. So if we look here, here's stratified squamous. So you can see they're all kind of flat. And then here you have your basement membrane. And then underneath that is connective tissue. So you have your basement membrane here. And then that's very obviously stratified, right? Because they're all stacked on top of each other. And they're also pretty obviously squamous. They're thin. Now, the reason that your skin is squamous and stratified is because the ones up here, squamous are really easy to produce because they have the least amount of volume. They're pretty cheap in terms of resources to make them. So you can make a lot of stratified squamous and just keep building them from underneath. Okay, questions on anything we've covered so far? Okay. Yes?
Is it possible to have a pseudostratified squamous?
I don't think so. I don't think I would ever see a pseudostratified squamous. Good question, though. I have yet to see them. If you find one, that'd be cool. Maybe I'll look it up. Okay, real quick, before we all get moving and packing stuff up. I know we have 60 seconds left. Okay, the rest of these slides, I haven't finished going through. So everything we went through today, make sure that you're aware of. I'm not going to be here Friday, so here's the plan. Tomorrow, we're going to go over connective tissue, kind of like we did today. And then Friday and Monday, you're going to do a little assignment activity going over epithelial and then connective. Cool? I probably will be here Monday, but I'll let you know for sure tomorrow. Cool? Excellent. If you have questions, bring them to me. If you had feedback about how this material was presented, I want to hear it as well.